Okay, class, we're back again. We were talking about the sarcomere between two Z discs. We described what actin is made of. We described this, which is the myosin. I said this was a sliver through right here. But on a three-dimensional, it's not just two actins. So let's go down here. Right here, I'm showing a side view. This would be, right here, is myosin. Here are the actins. I told you it'd be 6 around there. So if we do a 3D, you can see that it's not just this sliver here that we have. It's not just the sliver we have, but it's actually a surrounding of this. So this is kind of what I wanted to show here. All right. Anyway, we talked about the myosin heads. Here's myosin. We said it's turned around. And so right here, I'm going to say that in this, that's that M line. Right there's an M line protein. The job of the M line protein is to hook, come here, is to hook these myosins together. Now, remember I said on each head, you had actin binding site, myosin binding site, and an ATP binding site where you would put ATP, and that would be on both heads. Okay? All right. Now, the way the contraction occurs is basically this. Come here. That when we get into the contraction, you'll see it. Myosin will attach to actin, myosin, and then the heads will flip in and they'll slide this in from both sides. So you attach, and then you grab and you flip your head this way, which will pull you in and that in. All right, so we're going back to the sarcomere again. Okay. We said the titan holds this. That's the Z disc. Now, under the microscope. Now, let's say we're under the microscope. All right. We have these different zones that we're going to see. Okay. Under the microscope right now, I'm going to say that we have an I band. So we're going to start with bands. An I band. The I band is the area where there's absolutely no myosin. The I stands for isotrophic. Iso means same. Trophic means to seek. So under the microscope, with the light underneath the scope, you wouldn't see as much obstruction of light in these regions because you wouldn't have the heavy myosin. So this would be from this area here all the way to over here. Now that means if that's the case, then this I band would have a Z line or Z disc right in the middle. It would be where there's no myosin. And see, there's that Z disc right there. So the I band is where there's only actin and no myosin. Okay? Now, then we go to the A band. The A band extends all the way from in a sarcomere of the myosin because it's anisotrophic, which means it does obscure light. It's not the same refractive index as the glass. See, the I band has the same refractive index of the glass, ISO. The A band doesn't because you have this obscuring myosin. So that's that. So that means right in the center here, now right in here where you have this A band, look what you have from here to here. But right there you also have some actin in that A band. So if I come here and take out all the actin, which would be from here to here, we can call that the H zone. Why? Because it has the heavy filament in it only. Heavy would be the myosin. So the H zone only has the heavy filament in it. only has the heavy filament in it. This is the M-line protein. Okay, so what we've done, we've looked at I bands, A bands, the H zone, the Z disc in this. And we said, unlike that you see here on a three-dimensional aspect, it would look more like this here. With this being the myosin, and we have about 6 actins around it. See right here? They're trying to show that. So here would be just actin, I band. Here would be the H zone. Remember, the H zone only had the heavy filament. Here would be the M-line protein, where you also would just have the heavy stuff. And this would be M-line proteins. The job of the M-line protein is 2 in nature. Go back. The M-line protein not only hooks the two pieces of myosin together, so bond, bond, bond. The other thing it does is to keep the myosin in registry, which means so it doesn't fall. So 2 things are keeping it from falling on top of the actin. One is titin, which you see here, and the other is the M-line. It's locked here, so this can't fall. So therefore, what this drawing is showing is this is that locking of the myosin so it cannot fall. And here is the A band. A band, remember, had both myosin and actin. So this is attempting to make this. So again, I band, absolutely no myosin. A band, the full extent in a sarcomere of where the myosin is. The H zone right here, where you see absolutely just myosin, which is the heavy filament, but you see no actin. And the M-line for M-line protein, which some also call meromyosin, M-line protein or meromyosin, hooks the 2 halves together and holds everything in registry. Okay? And what happens in the contraction is these hockey heads grab on and pull in from both sides. Which then would cause the muscle to shorten because you would pull this in. So if you look at it then, you say, well, what gets smaller as you pull in? That would be the H zone. Because if I pull in from both sides, then I'm now introducing actin into the H zone. So the H zone gets smaller. The H zone gets smaller when you do the muscle contraction. So study that heavily because you need this anatomy to understand this physiology. Okay, let's go further. So we talked about the thick filaments run the entire length of the A band. We talked about all of this. There's nothing really for me to say. I'm not ready for that there. Okay, we've already done that. Okay, we talked about the ultrastructure of the thick filament, which is this here. The heads and everything. See, in other words, myosin tails are hooked together. The myosin head contains 2 smaller, lighter polypeptide chains. So up here, not only do you have this, but a couple of other polypeptide chains up in there too. Okay. And it has a binding site for actin, a binding site for ATP, and the ATPase enzyme on each head in one myosin molecule. All right, see what else we got. The ultrastructure of the thin filament, remember, let's go to the thin again, just being sure about it. The thin filament is made up of G actins. We polymerize them like 2 pearl necklaces. That's F, okay? The F actin consists of the globular actin units. Globular bears active sites for the myosin head. So let's go back to that picture. And we said that on every G actin, there's a myosin binding site. On every G actin, there's a myosin binding site. Then we came to tropomyosin, which is this ribbon here. This ribbon. And troponin. And the troponin had three components, an I, C, and T. The I hooked to actin, the C calcium when available, and the T tropomyosin. Okay, so this is what we're looking at here. Okay, now you see right here on this head, you have a couple of other polypeptides. So on the head, not only do you have the long one, the heavy chain, you also have two light chains, and that's what's being discussed here. Two smaller light polypeptide chains there. That's what's being discussed there to show that. Okay, and you interdigitate that, and now we have this right here. Okay, so that's what you're looking at. All right. So again, let me go to this picture here. We talked about myosin, myosin, okay, which is the M-line protein. That's another name for it. Titin, G-actin, okay, tropomyosin, troponin, alpha-actinin, nebulin we really didn't talk about. So it's a Z-stitch protein that assists anchor. I'm not going to get into that. So we're just going to stay with the ones we have here. The alpha-actinin, which holds here, and so forth. So study this. Look over, and study that. Because now what we're coming to, and here's another picture to show everything. The tropomyosin, the actin with the myosin binding sites and everything. Okay. Now we come to the sarcoplasmic reticulum. Okay. So now I need to go to a muscle cell inside. The sarcoplasmic reticulum is really the smooth endoplasmic reticulum in skeletal muscle. The sarcoplasmic reticulum is a maze of channels. The job of the sarcoplasmic reticulum is to store calcium, is to sequester and store calcium. The job of the sarcoplasmic reticulum is to sequester and store calcium. The sarcoplasmic reticulum is a maze, but it comes to an end point and swells up. It comes to an end point. Now, the lumen of the sarcoplasmic reticulum is called a cisternae. I brought that up before as a word in the cell chapter. Cisternae is a Latin word for box or chest. So inside here would be a lumen. And at the end of that lumen would be the termination of the box or chest. So the terminal cisternae. But remember, it's a maze. So coming from this side is this. Now, they're not two it's one long sarcoplasmic reticulum that just kind of wraps around. So there would be some over here, some here, and at the end of each of these blind loops of one maze of sarcoplasmic reticulum is this terminal cisternae. Now, right in the middle of the terminal cisternae is something we're going to talk about known as the T-tubule, which is at a transverse angle of the longitudinal axis of the muscle, a T-tubule we're going to see. So this terminal cisternae, that terminal cisternae, and this T-tubule form something we call the muscle triad. The muscle triad is the T-tubule plus two terminal cisternaes. The T-tubule plus two terminal cisternaes. That's the muscle triad. So what again does the sarcoplasmic reticulum do? It sequesters and stores calcium. Okay? That's the sarcoplasmic reticulum. Okay? All right. Let's see what we have here. The T-tubules are indentations of the cell membrane inward. The T-tubules are indentations of the cell membrane. It's like taking a balloon and poking your finger, but don't bust it. Okay? And it's associated with this pair of terminals which gives you the muscle triad. Okay? So there's that picture again. Okay? There's the triad. Okay? We'll talk about that. So we're going to close right here.